So in this lesson, we're going to look at exactly what happens to the genetic material during mitosis, but also what mitosis is used for in biology. So looking at the cell cycle as a summary, the first part is interphase, which we should have already covered. Hopefully you've seen my video on that one, where we look at the three phases of interphase, G1, S and G2, which is pretty much all about preparing the cell for cell division. The next part is mitosis, and this is what we call nuclear division. This is where we're going to divide the DNA. The DNA was all copied during the S phase of interphase. So now we've got two copies of the DNA, we need to equally split that in and form two new nuclei before the final stage of cell division and the cell cycle, which is to do cytokinesis and actually split the cytoplasm. So mitosis is split up into four smaller phases called prophase, metaphase, anaphase, and telophase, or PMAT for short. So let's start off with prophase. Now in prophase, the centrioles, they begin to move to the opposite poles of the cell. The nucleolus and the nuclear envelope break down, disintegrate, releasing the DNA, and the duplicated chromosomes become really clearly visible as two sister chromatids joined by a centromere in the middle. In metaphase, those centrioles that move to the opposite poles, they form a spindle out of microtubules, and those microtubules join the centromere of those sister chromatids. Okay, and once they've joined from either side, they can push and pull those chromatids so that they line up perfectly down the equator of the cell, or the metaphase plate as we call it. In anaphase, those microtubules pull and they split the centromere so that those sister chromatids come apart, and now they're back to being just chromosomes, and they get pulled to the opposite poles of the cell using ATP that was mass-produced during interphase. At telophase, the spindle can break down now, it's done its job. The nuclear envelopes reform and the nucleoli reform. The chromosomes can become less dense and just go back to being chromatin. And we've formed two separate nuclei with identical chromosomes in each one. So after mitosis, we need to do cytokinesis. This is where we split the cytoplasm to form two new cells. And it's slightly different in animals and plant cells. In animal cells, the plasma membrane is pulled inwards around the equator to form what we call a cleavage furrow by something called a contractile ring, which is made up of actin and myosin filaments. In plant cells, we need to form the new cell wall down the equator, and that is done by having what's called a middle lamellae, and then building cellulose either side of that to form these cell walls. And then that middle lamellae can disappear, and you end up with your two new cells. So mitosis is the most common type of cell division, and it has various uses: asexual reproduction, growth, and repair. So let's think about asexual reproduction first. Now, asexual reproduction involves just one parent. There are no males and no females, and that one individual can reproduce itself exactly. Genetically identical copies. These are clones, okay? And they do this via mitosis, all right? Now, that's great because it's simple and it's fast, but all the offspring are identical. There's no variation, which does make them vulnerable to changes in the environment. There's various different ways of achieving asexual reproduction via mitosis. You can do budding, which you see in yeast, where a small part of the cell starts coming off like a bud, and that will grow into a clone of the parent cell. Binary fission in bacteria, where they split perfectly down the middle to form two new identical cells. Sporulation, where fungi and some plants like moss reproduce by spraying asexual spores into the wind. Regeneration, which you can see in some animals like the axolotl, where they can replace whole body parts, or even sort of fragmentation like starfish, where they lose a limb and that can grow into new adults. Vegetative reproduction, this occurs in flowering plants, which then a part of them grows and becomes a new plant, like a runner in a strawberry, or bulbs, or tubers in potatoes. And then you've got the artificial ways using cuttings and micropropagation, where we can take small fragments of plants and remove them and grow them into clones of the parents. Growth is another major use of mitosis in biology. Growth is a permanent increase in the number, size, or mass of cells, and the best way to do this is via mitosis. We all started life, remember, as one single cell, a zygote, and now you are billions of cells, all produced from that first cell, and then done by mitosis after that. Organisms grow in different ways. Animals stop at maturity when plants continue throughout in regions called meristems. And finally, repair. Even if you are fully grown, your cells are still dividing by mitosis for repair. Now, you lose cells all the time. You've just lost 40,000 skin cells in the last 60 seconds of this video. They need replacing, and tissues become damaged, and they also need replacing all the time.